Welcome to the Witness History podcast from the BBC World Service. In this programme, first broadcast in 2010, Louisa Dalgo is telling us the story of two scientists who published a paper outlining their remarkable new discovery, the structure of DNA.
April the 25th, 1953. Nature. We wish to suggest a structure for the salt of deoxyribonucleic acid, DNA. This salt has novel features which are of considerable biological interest.
With such a memorable understatement, more than half a century ago, the two now most famous biologists of the 20th century, the American Jim Watson and the Briton Francis Crick, published a short paper in the scientific journal Nature describing their discovery. And what they'd found was the structure of life itself.
We saw, that was all one Saturday morning. I thought, oh boy, that's the answer. So the answer was very, very simple.
It was overnight we had the idea, and it only took a few days to build the model. We actually built a couple of models.
What they'd found was the now famous double helix, the molecular structure of DNA, the chemical substance out of which our genes are made, and how living things reproduce themselves.
We went from really not knowing where we were until a total answer all within a couple of hours or an hour or so. I thought, gee, this is beautiful. And, could it be this true? Because it was just a pretty structure. And we said, it's beautiful. You could describe it in a small amount of space. The whole essence of the thing, didn't take much to comprehend. And so that's why, our paper to Nature was, a page-long paper. It was tiny paper.
James Watson and Francis Crick were working at the Cavendish Laboratory at Cambridge University. A young lab assistant there, Mike Fuller, helped them to build the model of what they discovered. A giant replica of the double helix, two strands coiled around each other like a gently twisting ladder.
Being a young 15, 16-year-old, I was the gopher. And I remember going around on a bicycle trying to find different shops where we could buy sheets of cardboard that they could cut up. Once they'd got the structure out, basically the ideas in cardboard, of course, we tried to translate it into metal. Then we ran into problems. We had to get down into the workshop, and there was great pressure on the members of the workshop to actually just cut the tin plate out.
At last the model was finished, standing 6 feet tall and more than 3 feet wide. The molecular biologist supervising Crick and Watson's work, Max Perutz, recalls the moment he saw it for the first time.
One Monday morning when I came into the lab, Watson and Crick asked me to come into their room, and there stood this fantastic model reaching from floor to ceiling of the double helix. To a chemist experienced in structure, it immediately looked right. I could see that obviously they had got the solution. It must be right.
Francis thought it was his duty to tell everyone as quick as possible, because it should really form the basis for further thinking about how genetics functions.
We were in a state of euphoria about it, but it was tempered by the fact we weren't quite certain it was right. And this Jim said in his book, that every time he heard me telling people what a marvellous thing it was, he would have a queasy feeling that he was making a very big mistake.
But despite these private doubts, the story goes the two young men repaired to the local pub to celebrate, announcing to all of those there that they just discovered the secret of life. Talking later, though, Jim Watson says they knew that what they discovered was the culmination of many other scientists' work. Like the scientist Rosalind Franklin, for example, who'd produced the X-ray diffraction photographs which had made their discovery possible. And they didn't regard it at the time as the breakthrough in modern biology that it was later to become.
We didn't think of it as, people are saying now, well, it was the most important discovery of the century in science. We didn't think that way. I think we were, both of us, dominated by all the great things that had occurred in physics and chemistry during the first half of the century. And now people say, well, it was was very important because of everything that's happened over the past 45 years when DNA is just taken off in a sort of like a rocket. It's going faster and faster. I don't think it was, intellectually that clever. We were just there at the right time. And I've told Francis if we were chemists, we would have, found it in 6 weeks at most. And I was a, ex-bird watcher, and he was a physicist. And so neither of us knew the chemistry that we should know. And so really, when you think back, well, why didn't a chemist do it?
Mike Fuller remembers that even among Crick and Watson's colleagues at the Cavendish Laboratory in Cambridge, there was not huge excitement.
When Francis and Jim published the paper, it caused quite a stir within the unit. I mean, there were only sort of 6 people within the unit at that time, but did not cause a stir within the Cavendish. One day I remember them all coming out onto the stairs and being demonstrated the slinky spring, walking down the stairs. That caused tremendous interest because they all went back into the tea room and spent hours discussing the mathematics of how the spring worked. That caused a lot more interest than Francis and Jim's publication paper.
What James Watson and Francis Crick had discovered one Saturday morning in Cambridge would become the symbol of life science. But it would take another 13 years before scientists, including Crick and Watson, were able to work out how the structure worked and how the hereditary information was built in. It wasn't until 1961 that the genetic code was finally broken, and in 1962, James Watson and Francis Crick and 3 others were awarded the Nobel Prize for their work.
In Stockholm today, 5 men are receiving Nobel Prizes, the highest honours that international science has to offer, for work that will eventually lead to the healing of sickness and the preservation of human life. They've been working out the structure of life, unravelling the living cell to build up a picture as clear and in its way as beautiful as Beethoven.
The fact that the structure was interesting is why we got the Nobel Prize. If we'd proposed a dull structure for which wouldn't have given you really any clues as to really how DNA was copied, I think it would have been a hard game to say that you'd have a Nobel Prize for work which wasn't very exciting. I mean, we worried that maybe the structure would be dull and that the structure would only be interesting when it was inside a cell, not when you took it outside of a cell.
Once somebody mentioned it to me, I suddenly realised, well, good heavens, it's just the sort of thing they give prizes for, you see. But then I can't have been very conscious because, the announcement's made, is it November each year? And they'd always make the announcement before I got round to wondering what it was going to be that year. You're always supposed to wait for the telegram, and not worry because there's a journalist hovering about from Sweden, as there usually is. There was a story we filled a bath with champagne, but that's totally untrue. It is traditional to serve champagne. We fill the bath with ice and put the champagne bottles in the ice, actually.
Francis Crick died in 2004. Dr. Jim Watson went on to help launch the Human Genome Project, which helped map all the genes in the human genome.
That was the Witness History podcast presented by Louisa Dalgo and